By profiling cells or profiling tissues, making gene expression measurements, we learn about the cells or tissues in a tremendously powerful way.
Today, we feature Dr. Ricard Sandberg for his breakthrough work using single-cell transcriptomics to advanced understanding of gene expression at the resolution of a single cell.
I've always been fascinated by the ability to measure, genome-wide gene expression levels, to go from, measurements of single genes you have to, pick out before in order to do sort of global gene expression measurements. That's been my interest since my start of my PhD or even my master thesis. So my lab is located at the Karolinska Institute in Stockholm, Sweden, and I'm a newly appointed professor there in molecular genetics. And I have a dual appointment with the Ludwig Institute for Cancer Research. The single-cell methods, as you know, has really had a tremendous development, and now we are very happy to just be part of this research development. Why the single-cell methods have really had a dramatic influence on the research community is that it really became robust enough that everyone could use it. And it's easy to see how each one's research interest could benefit from having the single-cell resolution. Yeah, there's a great need to lower all kind of losses of RNAs, from when you harvest the cell until you sequence it, and by that, improving the overall sensitivity. So we can sequence more RNAs in the end of the ones that were present in the cell. And I mean, it would be great for the whole community, and it would, in particular, it would be valuable for cells that don't have so much RNA, or for organisms that have small cells with less RNA, and it will open up for analysis of these organisms and cell types. So what would be really nice is if we could somehow collect the single-cell transcriptome data that comes out in a manner that one can sort of have a curated resource of the gene expression profiles that sort of make up all the cells of our body. But this is tricky. It's not easy because we are using multiple different protocols for making single-cell RNA sequencing, and it's not straightforward how to combine them into a common sort of repository. One of the actually major challenges that have not been tackled enough or solved is how do we, in the best manner, get out single-cell suspension from intact tissues without hurting the cells or perturbing the cells. I mean, so far each tissue is sort of its own problem. How do we best dissociate out a tissue and get a single-cell suspension that one can analyze with single-cell transcriptomics? So I think that needs to improve. I think the method itself we could really improve in various ways. I mean, the most obvious perhaps is to make it more general for all RNA molecules in the cell, to allow us to profile microRNAs or other non-polyadenylated RNAs, for example. Of course, there's still room for improvement to make it in general better. I mean, we still lose more than half of the RNAs in a cell, and of course we would like to improve that. And I think the better the assay becomes, the more biological subtleties we can tease out, and there might be new discoveries there that we just cannot see yet. It has been, truly remarkable. It has, in just a few years, the single-cell methods have really spread out to the whole research community. I mean, if we go back to 2010 or 11, it was very difficult to make single-cell transcriptome, measurements. It was very expensive. Only a few labs can do it. But today it's just exploding. It's spreading to all aspects of science, and it's really remarkable. And it's to the point where it gets so easy to do it and so powerful that there's a need for it almost everywhere. So it's really taken off the last few years.
Innovations in RNA-Seq are speeding the journey to previously elusive answers. That's good science.